Hi, this is Dr. Kurt Wohler for Sunrise Functional Medicine. The title of this short video is Child with Autism and Alternating Hemiplegia Genetic Disorder. Here's the disclaimer for this information, understanding that this video and its contents are for educational purposes only. So the clinical history of this child involves a young child with a primary diagnosis of something called ATP1A3 alternating hemiplegia of childhood. Now, they also had an autism spectrum disorder. Now, ATP1A3 is a rare genetic disorder that adversely affects the exchange of sodium and potassium across the nerve cell membrane within the brain and nervous system. And this essentially disrupts the electrical chemical gradient that's necessary for normal nervous system function. So this is just a diagram of ATP1A3 at the neuronal synapse. And as you'll see, this particular area, or this particular what's called ATPase, that is involved in the exchange of sodium and potassium is compromised in these particular children. Now, a couple things to understand about alternating hemiplegia of childhood. Again, it's a rare neurodevelopmental disorder. It is characterized by repeated episodes of weakness, sometimes paralysis that can affect one side of the body. Many of these kids also develop seizure problems. They can have muscle stiffness, posturing problems, abnormal eye movements. And so the severity can be different for different kids. So they often have delays in developmental milestone development or formation as well, which was the case in this particular child, although this particular child did not have seizures. Now, there was an organic acids test that was submitted, and a couple things that I want to note are very important with regards to this particular case. The first two markers, acetoacetic and ethylmalonic, are linked to fatty acid metabolism imbalances at the cellular level. And often what's done in these particular cases is to administer L-carnitine as a particular supplement to help aid in fatty acid metabolism within the mitochondria, but the L-carnitine also has some antioxidant properties. The 3-methylglutaric is a marker that can be high. It often can be helped with antioxidants as well. CoQ10, that marker 3-hydroxy-3-methylglutaric, is elevated, indicating a CoQ10 deficiency. CoQ10 is a very important nutrient within the mitochondria, particularly the electron transport chain. Elevations of pyroglutamic indicate a glutathione deficiency. So if you keep in mind that a glutathione deficiency can compromise cellular function, particularly with regards to mitochondrial activity, well, we always have to ask the question, what's causing, or what are multiple factors that might be leading to a glutathione deficiency? But we know that the implementation of carnitine, antioxidants, CoQ10, and even glutathione can be beneficial. Now, the 3-methylglutaric acid deserves some particular attention or special mention here. So it turns out that 3-methylglutaric and another marker called 3-methylglutaconic, which is actually misspelled on this slide, are often elevated on organic acid tests when there is some type of problem in the metabolism of L-leucine amino acid. So L-leucine funnels through this particular biochemical reaction, and at the end it forms acetyl coenzyme A, which we see at the bottom of the slide. And acetyl coenzyme A, it can then enter the Krebs cycle of the mitochondria to help engage mitochondrial activity for the production of ATP. So there can sometimes be nutritional factors that might contribute to elevations of 3-methylglutaric or 3-methylglutaconic, and there can also sometimes be other mitochondrial imbalances as well as certain genetic problems. So there are multiple causes of elevations of 3-methylglutaric and 3-methylglutaconic acids seen on the organic acid test. Essentially, there's something interfering with leucine metabolism, which could then compromise mitochondrial function downstream. Now, in many of the genetic defects of the leucine metabolic pathways, the levels of these markers are often significantly high, particularly the 3-methylglutaric, usually above 10 millimoles per mole. Now, in this particular child's organic acid test, they were sitting at a 1.24 millimoles per mole. So it wasn't exactly consistent with a primary genetic disorder within this particular pathway. However, severe manifestations of defects in leucine metabolism can lead to speech delay, psychomotor delay, metabolic acidosis, often seen as elevations of lactic acid, poor muscle tone, musculoskeletal weakness. So there's a spectrum essentially that can happen in various people and various children who are dealing with mitochondrial problems or other metabolic imbalances. Now, an additional test was done called a mitoswab. And a mitoswab is a buccal sample that tests for specific points within the mitochondria for their activity, specifically citrate synthase, which is the first enzyme in Krebs cycle activity, and then complex 1 and complex 4. And I'll show you a diagram of this here with the next slide. What you'll notice in this particular child, the activity of citrate synthase, of complex 1 and complex 4 were normal. So that's great. That all appears to be working fine. So on the right-hand side of the slide is a typical mitochondria. So in the high metabolic organ systems of the body, the brain nervous system, the cardiovascular system, the musculoskeletal system, each individual cell in those systems can contain thousands of mitochondria. And the mitochondria produce large amounts of a chemical called adenosine triphosphate, or ATP, which is the cellular energy currency that's used by the cells of the body to make things work. The top image on the left is what's called the citric acid cycle or the Krebs cycle. The bottom image on the left is the electron transport chain. And the electron transport chain is embedded within the inner mitochondrial membrane within the mitochondria, essentially. And there are different protein complexes that essentially transport electrons from one complex to the next, and the end result is the production of ATP. So this was an image or the test example from the mitoswab looking at complex 2 and complex 2 and 3. It's a combination of complex 2 and 3. And you'll notice that complex 2 is normal. So we have normal complex 1, normal complex 2 activity, normal complex 4 activity, normal citrate synthase activity. But you'll notice the combination of complex 2 and 3 is low. And so we're not getting adequate flow of electrons down the electron transport chain. So even though complex 4 is working normally, what's happening upstream from that is not working normally. A couple other things to consider in these mitochondrial scenarios. Complex 1 is the largest protein of the electron transport chain. And the largest protein at complex 1 is also the greatest point of potential leakage of electrons into what's called the matrix of the mitochondria, where we have a lot of oxygen. And when we leak electrons abnormally into that space, we can generate reactive oxygen species, essentially these oxygen radicals that then further damage mitochondrial function. It turns out that complex 3 is the second most common point of electron leakage. So if we have deficits at complex 1 or deficits at complex 3 in its function, we're going to be leaking electrons into the matrix of the mitochondria, potentially generating increased reactive oxygen species, which then further damages the mitochondria to make ATP. Another interesting aspect in this particular situation with this child was there was a Intellex DNA test done. Now, Intellex DNA is a company that provides genomic testing. They have different types of profiles. This was what was done on this particular child was a neurodevelopmental profile. And the neurodevelopmental profile is very appropriate for children with special needs, autism, etc. And there's a lot of markers in that profile that look at mitochondrial function. And one notable genetic variant that was detected was of something called the folate receptor 1, folate receptor, or FOLR1. It turns out that this particular folate receptor is the primary receptor in how the brain draws in folate into the brain and nervous system. So essentially, folate attaches that receptor. It's then carried across the blood-brain barrier and then deposited into the brain. That process requires ATP, that same ATP that's made from mitochondria. And so this particular child actually had one mutation in the gene that oversees the control of that particular receptor. So some of these kids actually have what's called cerebral folate deficiency. This would not be absolute diagnostic of that, but it does suggest that there is some problem in the transport of folate into the brain and nervous system. This was picked up through the Intellex DNA report. So there's a lot of things that you can do in these particular circumstances. In this particular case, the implementation of a product called Spectrum Needs, which is a multivitamin, multimineral, and antioxidant, often very helpful for children with mitochondrial problems. It contains L-carnitine, it has CoQ10 and many other nutrients. Resveratrol has antioxidant properties. Sulforaphane also has antioxidant boosting effects. Both are very beneficial for mitochondrial imbalances, vitamin C as well. And then magnesium is often another useful supplement for supporting mitochondrial function. In fact, we need magnesium within the mitochondria to help preserve and protect ATP that gets produced. Another thing that was implemented is folinic acid. Folinic acid is a special form of folate that's often appropriate when we have some type of folate receptor problem or a suspicion of a cerebral folate deficiency issue occurring in special needs kids. If you'd like to get more details about this particular case, you can go to my Substack, drwohler.substack.com, and you can read more about it. And I have other information on my Substack as well. For information about our consulting practice, you can go to our website, mysunrisecenter.com. You can always call or email us. The email is scmedicalcenter@gmail.com. Again, I'm Dr. Kurt Wohler for Sunrise Functional Medicine. Thank you.